This is chapter 2 of the video pamphlet on agonist efficiency. In chapter 1, we defined the terms efficacy, affinity, efficacy, and efficiency in terms of the binding energies to the closed and open forms of the channel, the weak binding to closed and strong binding to open. Those are those two blue lines shown to the right there, with affinity being the lengths of the lines, efficiency being the difference between the lengths of the lines. Efficacy being the difference between the lengths of the lines, and efficiency being the ratio of the lengths of the two lines, or 1 minus that ratio. So affinity can be anything, and efficacy can be anything, as we showed previously, but efficiency turns out to be very patterned. And that's what makes it so interesting. It's nice to know to be able to measure the efficiency of one agonist versus another, but the real value of efficiency comes from this next section, which is its patterns. And there are 3 patterns in particular that I will talk about. So I'm going to start with an unexpected experimental result. This was made back 10 years ago in 2012 by Snail Jady and Prasad Purohit. And that is that the two binding energies are linearly correlated. So we measured KDC from single channel kinetics, and we calculated KDO, the strong binding to the open state, using the green equation, given our knowledge of L0, the unligated gating constant. We had just learned how to measure that. So when we made this plot, we were surprised to see that these two binding energies were so tightly correlated over a wide range of agonists, from very weak agonists, very low efficacy partial agonists, to a very high efficacy and high affinity full agonist, namely acetylcholine receptor. So this set us for a loop. We didn't know what this meant. The reason we were surprised is we knew that the binding to O would be stronger than to C. That means to the right of this dashed line. But we had no idea that it would be so tightly, there would be such a tight linear relationship. And moreover, with a slope that was 0.5. That troubled us because we didn't know why we would get exactly 0.5. So we thought it was an artifact or something for quite a few years. We sat on this. We didn't know what it meant. And it took a while, but now that we know the equation for efficiency, which was shown in the last chapter, this pink equation right here, we know that all of the agonists have the same efficiency. The slope of this line is log KDC over log KDO, and that is constant for all of these agonists in the series. So the agonist, this is the first pattern, agonists can belong to an efficiency class. So, and within a class, all the members have the same efficiency. So what that means in terms of our energy landscape is that the lengths of the lines and the difference between the lengths of the lines can change. That's affinity and efficacy, but the slope of the line stays the same, the ratio of the two lines. So that's the definition of an efficiency class. So that was a surprise. Now, what can we do with this? Well, we can combine the green equation, which is from the thermodynamic cycle, and the pink equation for efficiency into a new equation, which is gives us the form of an efficiency plot. And that is that if you combine these two equations, you get the blue equation right here, which is the equation for a straight line. Y equal mx plus b, where y is log L2 and x is log 1 by KDC. And simply the way you do this, this is the green equation for two equal binding sites. What you do is you solve this equation, the green equation for KDO, and then take the log, and then you solve the pink equation for the log KDO. Then you set the two equal and do some rearranging, and you end up with a straight line. This 2 in the blue equation for the efficiency comes from the two binding sites. It would be 1 if there was one binding site. So from the slope of the efficiency plot, you can calculate efficiency. And the y-intercept gives you the unliganded gating equilibrium constant. So it's a very useful plot because the inputs of this plot are very simple elements of the dose response curve. You calculate efficacy directly from max, that's log L2. And from the EC50 and the efficacy value, L2 value, you calculate affinity, the KDC. So the inputs are simply max and EC50, and the outputs are these very interesting parameters, agonist efficiency and intrinsic gating. And as I said earlier, intrinsic gating is the same for all agonists. So once you measure it once, you're done forever for that receptor type. And agonist efficiency is there are classes. So there are many more efficacies and there are many more affinities and efficacies than there are efficiencies. So you've really condensed things down by measuring efficiency. So efficiency and L0 are very interesting and useful parameters. And you can get them very simply from dose response curves from using this equation for an efficiency plot. That was first described in this paper shown down below. So here's an efficiency plot for a bunch of acetylcholine agonists. Some of them are shown on the right here. For each one of these points in the plot is a complete dose response curve where we measured L2 and KDC and put it on a plot. And you can see it does fall on a straight line for all of these agonists. They have the same efficiency, which turns out to be 51%. And that's true for agonists, again, that have a very wide range of affinities and efficacies. And also, they have different structural scaffoldings. So ACH, nicotine, and DMPP don't exactly look like each other, but they all fall on this same plot. So the same efficiency applies to agonists having different scaffolds. We did this for a number of for about 20 different agonists, and some of them fell on a line having a different slope, which gave an efficiency of 42%. And those include epibadidine, as I showed you in the dose response curve in the last chapter, but also epiboxidine, anatoxin A, azobicycloheptane, which is just the head group here, cytosine. All of these agonists have this bridge nitrogen, but they're also in this group are tetraethylammonium, which does not have a bridge nitrogen, and tetramethylphosphonium, which doesn't even have a nitrogen in there. So all of these fell along a line having a slope of 42%, and 3 agonists fell on a line that had a slope of 54%. And again, I will show you that this is a significant difference. The efficiency is an incredibly precise measurement, I think just simply because it's a ratio of 2 logs. But even a small, a 10% error in the EC50s does not translate into a significant difference in efficiencies. So 2 things to point out here. Number 1 is the bottom one, efficiency classes. Each of these lines represents a class. They are discrete and precise. Discrete meaning that they're not just scattered all over the place. They're very closely adhered to a line, as was the case with the surprising result number 1. And number 2, they're precise. This 54%, 51% are quite different. And the second thing to point out is they all project, these 3 lines were fitted independently to a straight line for the efficiency plot equation, and they all gave the same y-intercept. And that gave the same log L0 as you would expect for this receptor at minus 70 millivolts, which is when these experiments were done. So there are distinct efficiency classes. There are multiple distinct efficiency classes. That's the second pattern of the 3 I want to point out. Now, there was one other agonist that did not fall on any of those 3 lines, and that was varenicline, and that's shown here before. This is a nicotine smoking cessation drug treatment named Chantrix, and we used the same L0 to calculate its efficiency, and we came up with an efficiency of 35%. So we are now in the process of seeking some other ligands that might fall on this in wild-type receptors. So all of these pertain to wild-type receptors. The wild-type binding sites for all of these. So there are 4 different efficiency classes. Although the 35% needs to be confirmed with multiple agonists. So I just want to show the agonists here, and I've circled them with a line of the color that relates to their efficiency: 54, 51, 42, or 35%, and Previously, we did an analysis that showed that number one, only the head group matters. So here, these are three ligands that have very different tail groups, and here's three ligands that have very same head group but very different tail groups, no tail group, and they all fall in the same class. So the head group is what determines the efficiency, and it looked to us like it was the size of the head group that was important. So the smaller ligands have higher efficiencies. With varenicline being the biggest because there are no rotation bonds here. The whole thing is a head group. So that needs to be confirmed with more extensive calculations and simulations, but that's our hypothesis that we put forth in this paper below. I'm not going to talk about that in any detail because we really don't understand the structural basis of efficiency yet. We're working on that. But what I want to say is if you look in a pharmacology book or a paper online, you see this statement all the time. There is no correlation between affinity and efficacy. And that is a true statement. So here are those data. Here's 9 different agonists that I picked out from that table plot shown before where I measure plotting the efficacy and the affinity in a log-log format. This is L2 and 1 over KDC, but you can see there is no correlation. You can get, for any efficacy, for a given efficacy, you can get any affinity, and for a given affinity, you can get very different efficacies. The point I want to make is that that is true only if you ignore the class structure of efficiency. Affinity and efficacy are correlated. They're very closely, tightly correlated within an efficiency class. So here it is. The circles are the ones that I showed previously, and you can see that in an efficiency class you get a tight correlation. So the idea that affinity and efficacy are not correlated only pertains when you ignore the fact that there is an efficiency. And efficiency is a new idea, so most people haven't taken that into consideration. So I want to move on from the efficiency plot and show something that can be a useful aspect of knowing the intrinsic gating and efficiency, which are the two things you can get out of an efficiency plot from EC50 and max. So here's some dose response curves. These are whole cell dose response curves that I showed at the end of the first chapter, and I've added one for carbachol in here. And it turns out if you know the efficiency in L0, then you can calculate EC50 from max and max from EC50. That's part of the equation for the efficiency plot. So here what we've done here is we've taken just max from each of these 4 agonists and calculated an entire dose response curve from just max and superimpose them onto data. This is not a fit to the data. This is just a superimposition of the solid line from just max. So a complete dose response curve can be calculated from the response to just one agonist concentration. If you know the maximum response, you can calculate EC50. You don't have to scan all different concentrations. And similarly, down below, what I've done here is I've taken these dose response curves, we've taken these and we've normalized them to have a maximum of 1, as is often done because you don't know the number of channels that are contributing to the response. And we took we fit these to Hill equations just to get EC50, assuming a maximum of 1. And we took those EC50s and then calculated efficacy values, maximum values from that. So efficacy, and you can see that the lines agree with the points. These again are not fitted. Those are just superimposed. So efficacy can be recovered from a normalized CRC. Even a literature value in a paper, you can get your efficacy out once you've measured the efficiency. Since L0 is the same for all agonists, once you measure it, you're done. And efficiency, there's only a few classes. So once you know the efficiency of the ligand, efficiency class of the ligand, which you might be able to even tell from structure, you can then regenerate the full dose response curve from either EC50 or max. And I want to point out here, this recovery from EC50 is very sensitive to the value of efficiency. So if we use 55% for tetramethyl ammonia, and it really is 54%, we use 55%, you get a good fit. But if you use 50% for the efficiency of TMA, you get a very poor fit. And indeed, this may be one way, by comparing these two curves, maybe one way you can estimate efficiency of your ligand. Is another way that might be pursued. So that's, if you know L0 and N0, you can use it to analyze dose response curves. Now I want to turn to mutations of the binding site. So this is one of the few structural pictures I'll show. This again is from the 5kxi.pdb, which just to show that the agonist at the binding site is surrounded by a cluster of aromatic amino acids. There's two loops called loop B and this loop called loop C, which I'll refer to later on. And the big dog in this is Y190. It provides about twice the binding energy as these other guys. This guy here on the complementary side, on the epsilon and delta subunit, really doesn't do anything, but it does contribute in the gamma subunit. So now I'm going to show you the effects of mutating these amino acids plus a couple other ones at the binding site on agonist efficiency. So this histogram is a histogram of efficiency values following mutations, and this is with ACH. So you recall that the ACH wild-type efficiency is where this arrow is at 51%. And you can see this is about 45 or so different mutants, and you can see that most of these mutants do not have an effect on efficiency. So all of the mutations at Y198 and do not change efficiency. It stays 50%. About half of the mutations that we measured at 93 do not change, and the mutation Y190F does not change. However, there's a significant population of mutations that increases the efficiency of ACH from 50% to 54%. These include everything at the tryptophan 149, which I can't go back, but 59, 149, and in particular 149A. If you get rid of that bulky tryptophan, the efficiency goes all the way up to 60%. One amino acid was mutation Y198 dropped the efficiency of ACH all the way to 35%. Now, we also measured 16 different mutant receptor combinations, mutant agonist combinations for mutations of glycine up in loop B at the top of that picture in the last slide. And all of these lower the efficiency of choline, which is a 51% agonist, here shown in black, or DMP and TMA, which are 54% agonists. Both of these went down to 42%. And one of them went down to 32%. I think that was nicotine on G153K, went down to 32%. This inset shows the efficiencies for each agonist. I want to point out one of these mutations, G153S, causes the disease congenital myasthenic syndrome. A congenital myasthenic syndrome, and it's possible that the reduction of efficiency of the neurotransmitter from 51 to 42% could contribute to that disease, although that mutation also changes affinity and efficacy, and it could be those are the key reasons that you have a disease state. But that needs to be investigated, I think, whether it's affinity, efficacy, or efficiency is what the primary underlying cause is. We also studied these two mutations, D200A, K145. These are residues at the binding site. I'll point them out to you in a second. And everything that we did, ACH and all of these four agonists were dropped down into the 33% range, had an average efficiency of 33% when we got rid of this D200, when we made it D200A. They all went 33%. And the K145A was very interesting because it dropped the efficiency of the 51% ligands down to 41%, but it didn't do anything to the 42% ligands. So this is very interesting, and this is something that we need to understand what's the structural basis of affinity. This is the substrate for what we're going to do to understand this. Uh, but for now, I just want to point out that we know these numbers, that these are the same values that we had in wild-type receptors for different agonists, 54%, 50, 42%, and 35%. These were the four classes that we had identified previously. So now I think we have four modes. I'm going to call them modes of binding site because then that the binding site modes are the same as the agonist classes. So classes are for wild type receptors, modes come from mutant receptors, and I think that this distribution of classes and modes is going to represent the binding site's barcode, or perhaps spectrum is a better word for it, because there's going to be some fuzziness to each of these lines. So these are the four modes that we have observed so far at alpha, epsilon, and delta, the adult type binding sites. And we've also measured just a few agonists for the gamma binding site, the fetal type binding site, where that extra tryptophan comes into play. And those guys were all, I'll show you those numbers in a minute, but those were all about 56%. So we think that the fetal site may have a different, we have to measure the varenicline and epibatidine on those guys. Those are just for TMA and ACH class agonists. So binding site modes, this is the third pattern. The first pattern is there are classes. The second pattern is there are multiple classes. The third pattern is the classes and modes are the same. I mentioned that it might be possible to calculate efficiency from PDB files, and this slide is an example of that. This is unpublished work by these people shown in the box, Dinesh and Durthy, MJ Singh, and Pooja Goyal, and it should be treated as preliminary, but I'm going to show it anyhow. Basically, what we did is we took this PDB file, which is a muscle-type nicotinic receptor complex with bungarotoxin, and we removed the bungarotoxin and docked in various ligands, did a preliminary equilibration, and then ran 200 nanoseconds of MD simulations. And these three plots shown up above are a principal component analysis of those 200 nanosecond trajectories, in which case we found for all three agonists, we were able to identify three different subpopulations called M1, M2, and M3. These populations are defined mainly by the fluctuations, the positions in the loop C's, the two loops, loop C and loop F. The initial part of the trajectory was M1 and the final part was M3, and we found this in all agonists. Here's M1, M2, and M3 for epibatidine, and M1, M2, and M3 for tetramethylammonium. So we took M1 and M3 frames and calculated MMPBSA energies, including enthalpy calculations, including entropy calculations, to get a free energy estimate of binding of ACH and epibatidine to M1 and M3, and we found that there was a general correlation between the M1 binding energy, here shown in black, and the low affinity binding energy, here shown in blue, from dose response curves. And there was a correlation in the M3 binding energy with a high affinity open state binding energy for the two agonists. So based on that, we assigned the M1 population as being the C binding site and the M3 population the O binding site. And we were therefore able to use the MMPBSA numbers to calculate the efficiency as 1 minus the binding energy ratio. And we got the following numbers from the MD simulations: an efficiency of ACH of 41 and an efficiency of epibatidine of 33%. So there's both good news and bad news. The good news is ACH, the calculated ACH efficiency is higher than in the calculated epibatidine efficiency, as was observed experimentally. The bad news, both numbers are off by about 25%, too low. There's more good news. The good news is that 41 and 33% are two of the modes, two of the classes that we observed, although not ACH was 50 and epibadidine was 42, but we do see a 33% mode for varenicline. But there's more bad news. The bad news is that the reason that these are low are for different reasons, that the ACH was low because the open state binding energy was too low. And in epibadidine, the efficiency was low because the closed state binding energy was too high. So we really don't understand what's going on, and we need to do a lot more experiments to verify that we might be able to calculate efficiency from structure. But if that were possible, it would be significant in two regards. And number one, you could calculate efficiency from the CNO structures that are going to become more and more prevalent as time goes on. And that would be a wonderful thing. You wouldn't have to express any proteins or do any wet bench experiments if we could show that this was true. And secondly, we'll be able to use these structures to try to decompose what's going on inside of fishing. What are the structural determinants of efficiency, which of course is a major question to be answered. And I'm just going to show here snapshots from the M1 and the M3 cap populations. In M1 showing that the orientation of the ligand is with the quaternary mean pointing away from the principal subunit and being distant from the key 190 residue. And in the M3, which is the open state structure, putative open state structure, the agonist has flipped its orientation and is now pointing its nitrogen towards the principal alpha subunit and is in close contact with the Y190, which we know is the principal determinant of binding energy. So these, by the way, are D200 and K145, which I mentioned earlier. So these are preliminary results, but they suggest that it may someday be possible to calculate efficiency from structure. So that's the end of this is the end of my second chapter on patterns. The binding sites belong to discrete efficiency classes. Binding sites adopt discrete efficiency modes. The distributions of classes and modes are the same. And our conclusion from that is that the binding sites adopt discrete CNO structures. That is, it's not like a rubber room, which the binding site can be anything. It goes to it starts and ends at preferred structures, pre-existing structures. So it's not induced fit, if you will, and it's more of conformational selection. And I think the O binding site is the key to understanding efficiency. So I think we'll be able to calculate structure and efficiency. And I just want to end by showing you a list of all the efficiency values that we have for different mutations and different agonists. And there's a lot of them. And down here, I'm showing you the four that I'm ending with the gamma subunit, where ACH, carbachol, TMA, and choline are all 55, 56%. So that's the end of the second chapter of the pamphlet. And the next chapter is going to have to do with the origins of efficiency.